Hi everyone. In this video, I'm going to be explaining the transport of sugars, which is in topic 9.2 of the IB biology syllabus. So firstly, what is active translocation? Translocation can be defined as the movement of organic compounds such as sugars and amino acids from sources to sinks. Let me just write that down. From sources to sinks. So a source is any region where the organic compounds are synthesized. So for example, leaves, which produce sugars during photosynthesis. Sinks are where the compounds are delivered to for use or storage, such as roots, seeds, and fruits. However, sources and sinks can vary based on the plant's life cycle. So for example, when it's a seed and it's still very much developing, the source would actually be the cotyledon inside of the seed because that's where most of the nutrients are stored. And the sink would actually be the leaves, which are still growing and developing and are not able to photosynthesize and produce their own sugars yet, right? So in this way, translocation can be bidirectional because the source and sink locations are variable. So sugars are always transported as sucrose, which is a disaccharide. And the reason they're transported in this form is because sucrose is soluble, energy efficient, and metabolically stable. So how does the transport of sugars actually work? To understand that, we must first understand phloem structure. So the phloem is basically made up of sieved elements, which are elongated cells that are connected together to form a sieve tube, which is what is shown here in the diagram. They have perforated end walls, meaning end walls that just have holes in them, called sieve plates, which enable the flow of sap between the sieve elements. So again, sap is basically just the viscous fluid of the phloem, which contains all of the organic compounds and nutrients. So the sieve elements also have thick and rigid cell walls to withstand the high hydrostatic pressure that's required in phloem transport. I'll explain that later in the next section. Their cell membranes also contain protein pumps that can control the composition of the sap if needed. The sieve elements also have a reduced number of organelles and no nucleus to maximize the space for translocation of materials. However, since translocation is an active process, meaning that it requires ATP energy, it's important to remember that the sieve elements are still living. They're not dead cells like the xylem. They're still very much alive. However, people often describe them as cells that are on life support, meaning they require a lot of support from their companion cells. So companion cells are connected to the sieve elements by a bridge or a passageway called the plasmodesmata. This allows the companion cells to provide metabolic support to the cells and for the cells to communicate between each other. So the companion cells also have adaptations which aid with this. For example, they have infolding plasma membranes to increase the surface area to volume ratio for faster material exchange. They also have many mitochondria to fuel the active transport of materials, because mitochondria, as we remember, produces ATP energy from glucose. So let me just write down the key main points. Sieve tube elements, or it's just sieve elements, have perforated end walls. They are thick and rigid. They have membranes with protein pumps. They have a reduced number of organelles and no nucleus. They're connected to the companion cell via plasma desmata. And the companion cell has high SA to V ratio due to inner folding membrane. They also have lots of mitochondria. Okay, I hope that makes sense. Let's move on to the structure of the roots and stems in a monocotyledon and a dicotyledon. Again, this is just something that we need to know how to do in this topic. So it's always important to remember that for the roots of the stems, the xylem is always on the inside and the phloem is always on the outside. As you can see here, xylem is inner, phloem is outer, phloem is outer, xylem is inner. A nice way to remember the root dicotyledon is that the xylem forms an X shape, and the xylem and the phloem together, as you may know, are called the vascular bundle. Other than that, this is just something that you need to memorize. Yes. So let's move on to phloem loading. So organic compounds at the source are actively loaded into the phloem sieve tube so that they can be transported. And this is done by companion cells in two different ways. So firstly, we have symplastic loading, which is via the interconnecting plasmodesmata, which remember is the bridge between the sieve tube elements and the companion cells. And then apoplastic loading, which is pumped across by membranes, meaning it's an active transport process. It uses ATP energy. Okay, so firstly, H plus ions or protons are actively transported out of the phloem cells by proton pumps, which uses ATP, remember, because it's actively transported. This means that the concentration of hydrogen ions builds up outside of the sieve element cell, creating a proton gradient. Because remember, there's a high concentration outside and a lower concentration outside. The hydrogen ions passively diffuse back into the phloem via a co-transport protein, which regulates sucrose movement into phloem. This means that in order for the hydrogen to diffuse back in, it must take a sucrose with it. And this is basically how the phloem is loaded with sugars, from the passive diffusion of hydrogen back into the phloem via a co-transport protein. And again, remember, the proton pump here is an active process because there's already a high concentration here and a low concentration here. So we must transport the hydrogen ions against the concentration gradient. So this results of the buildup of sucrose within the sieve tube for transport to the sink, or sinks plural, because again, there can be multiple. So how does this actually happen? It's done by a process called mass flow. So the sap solution is actually hypertonic, meaning it has a high concentration of nutrients like sugars and amino acids. So this means that water will actually osmose or move via osmosis into the phloem from the adjacent xylem cells, from lower to higher solute concentrations or higher to lower water potentials. It's the same thing. So if you remember, the xylem and phloem are actually right next to each other. They're both in the vascular bundle. So it's very easy for water to just move from its low solute to high solute regions. So water is incompressible, meaning that its volume cannot be decreased, meaning that it starts to build up in the phloem, causing a very high hydrostatic pressure. And this forces the phloem sap to move towards areas of lower pressure, which is towards the sink. To relieve some of this pressure, it has to go away from this really high pressure area. So at the sink, the solutions are unloaded by the companion cells, as you can see here, and this causes causes the sap solution to become increasingly hypotonic or low solute concentration. And this means that the water will begin to move back into the xylem. So it's kind of like a cycle in which water is sucked in here due to the high solute concentration and then sucked back due to the transpiration stream. Yes, this means that phloem sap always moves down the hydrostatic pressure gradient from the source to the sink, because again, the source will always have a higher hydrostatic pressure than the sink, because this is where all the sugars are naturally produced, and hence this is where the concentration of sugars is the highest. So the final bit of this unit is translocation rate. So one way to measure this is using an insect called aphids. Aphids have stylets which can pierce a single phloem tube and cause the sap to flow out without affecting the plant. They can be used to analyze the sap chemical composition as well as its direction of flow and rate of transport with labeling of radioactive carbon dioxide. So the main factors affecting transpiration rate is rate of photosynthesis, rate of respiration, rate of transpiration, because this determines how much water actually enters the phloem for mass flow, as well as the diameter of the sieve tubes, because this affects the hydrostatic pressure and may differ between different plant species. Again, photosynthesis and respiration is just because of glucose needs. So if there's a high need for glucose in the roots, then and more like sugars are being produced by photosynthesis, then obviously it would move towards the root at a faster rate. So yeah, that's all that's in this unit. I hope it made sense. It should be a fairly straightforward unit as long as you understand the processes and the reasoning behind everything that's happening. I hope this helped. Bye.